Now let's look at some of the some of the players in the cell division. One of the most important players is chromosome. Let's look at its structure. Chromosomes are generally not visible in cells which are in G1 phase. Chromosomes are unwound, they are loose, and they are not present as discrete structures. When the cell decides to divide, chromosomes become condensed and they become visible under the microscope. Chromosome basically has two components: DNA, the genetic information, and protein that is serving as a scaffold around which DNA is wound. I'll show you a photograph in a second. Basically, the length of DNA in our cells is about 2 meters, and the size of nucleus is 500,000 times smaller. So in order to put DNA, such a long DNA, in such a small space, we have to wind it around. Just like you can wind thread around a spool, and even though the thread is very long, if you wind it around a spool, it will become a condensed structure. So let's look at the structure of chromosome. We have the DNA, as you can see on the screen. It is wound around these little beads. These beads are made up of proteins. These special proteins are called histone proteins. There's a special arrangement to it. There are histones, 8 molecules of histones, they form the bead, and little more than 1.5 turns of DNA is wound around each of these beads. Another type of histone called H1 basically clamps the structure. The DNA is wound around these histone molecules, and there is a certain part of DNA that is not wound around these histones, is called the linker DNA. The structure, the protein, 8 protein molecules of histones, and the DNA basically is called a nucleosome. These histones are positively charged. As we know, DNA is negatively charged because of the phosphate group, and in physiological conditions, these histone proteins have a positive charge which allows DNA to wrap around these molecules very tightly. Now, let's look at another important component of cell division: the centrosome and the microtubules that originate from the centrosome. There are two types of microtubules: the kinetochore microtubules that go and bind the chromosomes made up of DNA and protein, and other microtubules that are not bound to the microtubules. These microtubules are attached to the centrosome, the organizing center, microtubule organizing center. When the cell division happens, we have to remember separate these two chromosomes, two sets of chromosomes, and force has to be generated. We have already seen that microtubules are scaffolds, or the substance along which motor proteins can move. Force has to be generated. The force is generated, we will see a video of it too. That force is generated between the microtubules. When the force is generated, the two centrosomes are pulled apart or pushed apart, resulting in two poles, both containing one centrosome each and a set of chromosomes. In the next stage, a membrane is formed between these two microtubules, these two sets of chromosomes and centrosomes, resulting in two different cells. Next, we will look at additional information about cell division.